From the University of Wisconsin-Milwaukee's Office of Research, welcome to Research Matters. Welcome back to Research Matters, where we spotlight standout research happening across campus and beyond. Today we're talking about fire blight. If you work with apples or pears, you know this disease. It's caused by the bacterium Erwinia amylovora, and it can devastate orchards. But a new study by UWM's professor of biological sciences, Ching Yang, published in the journal Phytopathology, offers a different, non-antibiotic way to manage it by disarming the pathogen instead of killing it.
Right. The team tested a set of small molecules called type 3 secretion system inhibitors, or virulence inhibitors. Quick primer. Many gram-negative bacteria use a type 3 secretion system, a little molecular syringe, to inject proteins into host cells and suppress defenses. If you shut that down, the pathogen loses its edge.
Exactly. The paper focuses on a compound called TS108, a derivative of plant phenolic acids. What's remarkable is that they didn't just do this in petri dishes. They ran multi-year field trials in 4 states: Michigan, New York, Oregon, and Connecticut, between 2015 and 2022. In some trials, they saw dramatic reductions in blossom blight, in a few cases approaching the performance of standard antibiotics. In New York, one related compound cut blossom blight by up to 96.7% compared with untreated trees.
That's huge. But this isn't just a spray and pray story. The researchers also figured out how TS108 works at the molecular level. They found that TS108 turns down the bacterium's main genetic control switch for its infection machinery, the system it uses to inject harmful proteins into plant cells. It does this indirectly by disrupting an internal regulatory signal that normally tells the bacterium when it's time to be aggressive. Once that signal is dampened, a whole chain reaction follows, and the secretion system never fully turns on. The bacterium is still alive, but it's effectively disarmed and far less capable of causing disease.
An important nuance is that TS108 appears to be species-specific. It suppresses T3SS gene expression in E. amylovora, but does not repress comparable systems in other bacteria. This specificity is significant because it reduces the risk of unintended impacts on beneficial microbes that may utilize similar secretion machinery.
Let's talk field results. In New York, TS108 performed on par with S-streptomycin, that's a commonly used antibiotic, for reducing blossom and shoot blight in some trials. In Oregon and Connecticut, it still helped, though antibiotics tended to outperform it. And in Connecticut, researchers also observed a strong synergy with a biological control yeast product called Blossom Protect. When the yeast was used together with TS108 or another phenolic compound, disease incidence was reduced more than when either treatment was used alone. This finding is important because it shows that TS108 can be used together with living biocontrol agents without inhibiting their activity, allowing growers to combine beneficial microbes with additional disease suppression strategies.
So where does this leave us? First, this is a proof of practice that antivirulent strategies, those that block disease mechanisms rather than bacterial growth, can work under real orchard conditions, not just in controlled environments. That's a milestone for plant pathology.
Second, the antibiotic resistance angle. Fire blight management has leaned on antibiotics like streptomycin for decades, and resistance has emerged in several regions. Antivirulence compounds like these lower the selective pressure for resistance because they don't kill the bacteria outright. That could help preserve antibiotics for when they're truly needed and reduce environmental and public health concerns tied to agricultural antibiotic use.
For the field itself, plant pathology, microbiology, and even chemical ecology, this study opens up design principles for the next generation of inhibitors. Medicinal chemistry can now ask, how do we make TS108-like molecules more potent, more stable in sunlight, and longer lasting on plant surfaces?
And timing matters. The authors point out that E. amylovora expresses the secretion system during epiphytic growth on flower stigmas, basically when it's living on the flower surface before invading the plant. That suggests the best spray window is during bloom, aimed at stigma colonization. Hit the pathogen when it's gearing up to invade, and you may prevent the cascade that leads to blight strikes.
For growers and extension specialists, there are practical implications. First, this looks like a new tool for integrated programs, especially where streptomycin resistance is prevalent or where there's a push to cut antibiotic use. Second, the combo strategy, pairing T3SS inhibitors with biologicals, seems promising. And third, cultivar susceptibility still matters. The compounds did especially well on some highly susceptible cultivars in certain years, less so on others. So we're not talking about a silver bullet, but a smart addition to the toolkit.
For the broader public and policy folks, this study speaks to how innovation in basic bacterial biology can translate into more sustainable agriculture. If we can scale anti-virulence approaches, we can reduce antibiotic inputs, protect beneficial microbes, and still safeguard yield and quality.
Let's also flag what's next. We need formulation work. Things like UV stability, surfactants, and adjuvants to help the compound stick and persist on flowers. We need dose optimization across cultivars and climates, and long-term monitoring to confirm that antivirulence agents really delay or diminish resistance in the field. And of course, regulatory pathways. These data are exactly what agencies look for, but more multi-location seasons will strengthen the case.
Bottom line, disarm, don't kill might be the new mantra for bacterial plant diseases. This paper shows it's not only biologically sound, it's agriculturally viable.
If you'd like to dig deeper, the paper is linked in the post description. It includes the mechanistic data, field trial tables, and an extensive discussion of timing and integration with other controls.
If you're faculty or a research lead, consider sharing it with colleagues in plant sciences, microbiology, and public health. It's a great example of bench-to-orchard translational science.
That's it for today's episode of Research Matters.
Until next time, stay curious.
Research Matters is produced